obviously call me Tom, but of course that sounds like a good idea. We do have the face shields, and maybe I could go with that. All right, so
Okay.
I'll be quiet. I'll be quiet. Look forward to Neal's talk here.
Okay, Fox. So it's 9 o'clock. So let's see. Okay. Good morning and good afternoon and good evening. As always, we have audience from Europe, from Asia, of course, North America, South America. So colleagues, thank you very much for attending this World River and Delta Series Source to Think webinar series. So today is the number 10 of 2021. So we welcome Professor Neal Blair from Northwestern University to give a talk about the generation and propagation of the storm pulse in the Fluvial system, particularly look at the POC. So before I introduce Neal, so this coming Friday, Hans Paul from our neighbor UNC Chapel Hill Institute of Marine Science, he will also talk about the impact of the tropical colonial, particularly the hurricanes, what's the coastal nutrient and the carbon loading and ecosystem response, so same time this Friday. And at same time next week, so the next week, we have Xi from Singapore, National University of Singapore. He will talk about the riverine sediment load response to climate change, particularly global warming, the high mountain area, and the Himalaya mainly. How the global warming, melting of the ice, increased sediment, and how the water discharge. And Justin from UK, he will talk about another very interesting river, the Amur River, is the Heilongjiang River. So that's not too much, study over this river. That's very interesting, between the border, China and Russia. And so please mark your calendar. So now, Neal is a professor in Northwestern University, and his research mainly focuses on behavior of the carbon. And so those could include the terrestrial, both terrestrial and marine environment. And his study, he has conducted field study in North Carolina, California, New Zealand, Papua New Guinea, Taiwan, and Amazon River. So Neal received his bachelor degree in chemistry in University of Maryland and also PhD in Stanford University. And so I didn't realize, even I'm a colleague for 4 years here in North Carolina, I didn't realize Neal also is a research associate in the NASA program, Extraterrestrial Research Division. So Neal, I didn't realize you are the alien searching group. So, but anyway, and Neal has many collaborations. As we mentioned back to earlier time, he working with George Yuan of Arizona State University, particularly focused on that organic carbon from extraterrestrial. And also David from David D. Marais from NASA, and later on working with Chris Martin, UNC Chapel Hill, and also Neal joined the AMSAT in early 1980s, led by Chuck and Dave, and also working many times with Bob Eller, particularly try to understand those Amazon shelf, and later on working with Neal, with Lonnie. And so also working with Lisa Levine and David Master. So I think, today Neal will give us talk about those kind of carbon signature all the way from terrestrial to the marine environment. So Neal, now you can share your screen and the PowerPoint.
Okay, how's that look?
That's very good.
Good. So anyway, thanks, Paul, for inviting me back home, however virtually. It's nice to see that some of the NC State crew. And also thanks for inviting me back to the Source to Sync community. I miss that a lot. I remember fondly, see if I can change the slide here, workshops that we had in NSF, part of the Source to Sync program. And one of the things I remember most vividly, I think it was Bill Dietrich and probably Gary Parker and a few others, who were sketching on the board this idea of what a storm pulse signal would look like as it moves down through the system, and how it would dissipate and what kind of processes would influence it, with the idea that when you get it in the marine environment and it gets recorded in the seabed, what are you really looking at? And so what I'm showing here is just a schematic or a figure from Brian Roman's article, which I suspect almost everybody's read multiple times. It's a really nice review article on source to sink perspectives, where he sort of talks about the peak of sediment moving down a system and what might happen to it as it propagates. It gets really interesting because it's more than the peak. The signal is more than just a sediment wave moving down a river. It's going to be interacting with the river corridor. And as this little diagram shows, some of the stuff's going to go off and reside for a while in a floodplain soil or something and then come back in. And so that has an impact on the signal too. So ever since then, I've just been thinking about, boy, wouldn't it be really great to do an experiment to look at how a signal propagates down through a river system or a stream system using organic carbon, using carbon. And why that would be the case is that carbon is an information-rich element. When you think of all the molecular forms that you could have that contain carbon, all the various types of biomarkers, and we'll talk about some of these in the talk, the elemental compositions, the isotope compositions, the fact that we have stable isotopes and radioisotope. There is so much information embedded in carbon. How could we use that to really study signal propagation and learn about the system? So Tom showed this figure last week. It's one of our favorite figures. It's a cartoon, it's a caricature, and as cartoons go, it exaggerates certain points to make a point at the expense of reality sometimes. But what's the issue that we're trying to make? We're trying to argue here that you could view material transport across the surface of the Earth as a bunch of reaction transport couples in which material, when it's residing in a soil, for instance, the organics are turning over, they're reacting. Then it gets moved downstream to another place. And then the clock gets partially reset when that material goes into a new soil or a new environment with a new ecosystem. But it never fully gets reset. There's always a memory imparted on this. If you've got systems, and we've sort of cast this in terms of active margins, passive margins, you could really just say small systems, small steep systems, and big systems, where you have a shortage of storage. The end result is that a lot of your organic types, whether they're fossil carbon from the bedrock or terrestrial carbon or marine carbon, tend to get layered on or added into the system with modest turnover. However, if we have plenty of time, and let's take the Amazon as the extreme end member, which is covering half of a continent. A particle coming out of the Andes, we don't know how long it takes to get out to the ocean, but it might be millions of years. And by that time, the organics have been recycled many times over. And so this figure on the left is from one of Lonnie's papers, which I really like it because it just tells you in an instant what we're talking about. These hypsographic curves, we're looking at relative height versus relative area. And what you note is for the larger river systems, the more passive margin systems, we've got these large areas that are fairly low gradient, which means we've got large reactor volumes where there could be a lot of turnover of organics. Whereas if we get to these smaller systems, there's really not a lot of place to hide sediment and store it. It happens. There's floodplains, there's debris tails, and what have you. But we don't think that the storage capability is anywhere near what we have in the larger systems. And so one of the things, there's been a lot of study done by us as well as other people like Bob Hilton, who you should look at his papers. He's done really nice mechanistic work on small mountainous rivers. And of course, there's been a lot of work done on large rivers from the large river perspective. But what I've been really curious about is what is happening if you zoomed in to a little tiny place in this reactor sequence. What would you see? We actually don't have a lot of information for this. So before I go there, I want to point out there's another problem that we're going to be trying to address with this type of research. And this has to do with the impact of humans on the carbon cycle, which we all know is a big topic. But instead of talking about fossil fuel combustion and CO2 emissions necessarily, we're talking about this lateral part of the carbon cycle where stuff's moving across the landscape. As it turns out, humans have moved about 50% of the landscape, have caused erosion to some extent on 50% of the landscape. And you can see this red line is tracking what are estimates of soil loss caused by humans relative to natural system. And you can see it's many times above that. If you were to put an average carbon load onto those particles, say 1 to 3% carbon, what you find is that potentially we've disturbed a flux of 1 to 3 petagrams of carbon per year, which is getting up to a big number. It's up there with the fossil fuel estimates. So the issue that comes out is that we don't entirely know what that disturbance means. How much of that carbon's going in the atmosphere? How much of it is going into the ocean? Well, we can say something about what's going into the ocean because people have done budgets of that. But what's going on in between? And so this is just an example. Just you might wonder, why do we care? Our greatest environmental disaster in the US, the Dust Bowl, was created by disturbing soils. We over-tilled them, and we end up losing a lot of the organic carbon. This is more than half of it in a typical ag soil back at the beginning of the 20th century, which removed the glue between the particles. And then we had a series of years of droughts from La Nina that end up just blowing everything all over the place. So this is an extreme case, but what it's illustrating is that the disturbance caused by land use, particularly agriculture, ends up causing us to lose carbon from the soils. So the question is, where does that carbon go? And so this diagram is sort of comes out of, it's my diagram, but it comes from information from Bob Mead's article where he was asking the question, where do eroded soils go? And he concluded that less than 10% of eroded soils actually make their way into the ocean. So this is something to think about, because what you're looking at coming into the ocean from the continents is a leak out of a very complicated system that potentially has a great deal of filtering capability. So you don't really know a priori what you're seeing coming into the ocean. So it behooves us to try to understand and break this system down. So Bob basically argued that you could find the eroded soils in 3 different places: on land, downslope traps at the base of hill slopes, floodplains, and lakes and reservoirs. Bob Stallard wouldn't put carbon numbers on some of these things as well. To make this an even more interesting problem is, originally when you look at diagrams like this of the agricultural soil, if you make the assumption that that carbon's going into the atmosphere, CO2, that's a very big number. It's 1/3 of the anthropogenic CO2 flux or more. And so it's always been assumed, or originally it was assumed, that land use, such as agriculture, was a net source of carbon to the atmosphere, not a good thing. More recently, people have been developing models that take a more holistic approach, and they look at the carbon exchange in these boxes, and they've actually come to the conclusion that it's possible for this system, the whole system, to be a net sink of carbon. In other words, erosion is good, at least from a carbon perspective. I don't think it's very easy, and in fact, I'm not sure it's possible to tell the difference. But what I hope you recognize is that this problem in the carbon cycle is a source-to-sink problem. It's something that we can take source-to-sink philosophies and ideas and apply this on land to try to understand what is happening to the carbon. So anyway, that's the other big issue that we want to look at. So what we're going to do is we're going to go and zoom in with our landscape microscope and look at a watershed that's about 200 square kilometers in size, and really look at how a signal is generated and then what happens to it as it moves, say, 10 kilometers or so, very short distance. Then we're going to expand our scale and go out to a 2,000 square kilometer scale to really address this issue that Mead brings up, is where does the eroded carbon go? And I just should note the 2,000 square kilometer scale is very similar to the small mountainous river scale. So we're going to see a compare and contrast a little bit between those two types of systems. So we're going to be looking at very low gradient systems in the Midwest. So I know all the marine types, hang in there, because ultimately we're going to try to push a little bit of stuff into the ocean. Okay, so the first place we're going to is Clear Creek in Iowa. And let me just tell you, when I was at NC State, the last place I ever thought I would be working would be a cornfield in the Midwest. I never imagined it. But you know, when you move to the Midwest, you do what the Midwesterners do. You play cornhole and go out and study cornfields. Anyway, so this little field site is actually by the University of Iowa. It's been one of these extensively studied sites. It's kind of like Cape Lookout Bight to North Carolina oceanographers. It's part of, or was part of, the Intensely Managed Landscape Critical Zone Observatory, which was one of the CZOs the NSF had sponsored for about a decade. And what we were able to do was the experiment I always wanted to do, and we couldn't do it because of logistics, was to set up sampling sites longitudinally so that we could track a storm pulse down through the system. And so these were auto samplers, so we could collect samples all through a storm, and then we could define the storm pulse and actually watch it through. So what we're going to do first is set the stage, look at the environmental issues that we think are going to impact what we see. We're going to look at the sources of the POC, the actors in this, and then we're going to create our story. We're going to try to knit all this together to create the signal and its propagation. And if you like this kind of stuff, we have two papers. One of my grad students, June Kim, had a paper come out with Biomarkers last year. And then literally our other paper, which is on the Clear Creek thing, was just published last night. And so you're looking at really fresh stuff here. Fresh carbon. Okay, so landscape, agricultural belt, surprise, surprise, lots of corn and soybean. So all the green and gold coloration here is the land, is how the land's been converted to row crops fields. They're all through the watershed. You can see them here and here, but definitely a higher concentration up in this region around South Amana. We have forest, we have a little bit of prairie, lots of pasture, a little bit of urban area, Coroville's down here, the university's down here. And so we have a good sense of land use. One of the things I want to point out here before I forget is corn. Corn is a C4 plant. It's a type of physiology. Most plants are C, we call them C3. Corn is a C4. A lot of people, for those of you not in the US, this is also called maize. What's notable about corn is it's enriched in carbon-13. It has a delta C13 around -10 to -13 per mil. Why do we care about that? This is equivalent to us going out and spreading a lot of carbon-13 all over the surface, except that NSF wouldn't have given us enough money to put this much carbon-13 out on the surface. Okay, so the farmers have done it for us, and so you're going to see in a few minutes how we use this as a tracer for surface processes. Valley geomorphology. This is the type of thing that actually the source to sinkers would be talking about, would be how the actual river valley influences what the signal generation looks like. So south of Mana, so let me first say that these little asterisks mark the channel, these little narrow areas. South of Mana, the valley is almost a V shape. And we know from experimentation, we know from modeling, that this really facilitates the connectivity between the hill slope and the channel. Okay, so for those of you not familiar with sort of the terrestrial geomorphology, we like to talk about connectivity. In other words, how well does something move from one surface and make it into the channel? So up at south of Mana, we really expect that there's really good connectivity for material from the surface to get into the channel. However, as we get downstream, we're going to more of a box shape. And what that means is that it makes it harder for stuff from the hill slope to actually make it into the channel in one swipe. In other words, it may move down to a point, stop, and then we'll have to wait for the next storm to move along again, so it hops along the surface. So we're starting to approach that little bit of hopping that we were talking about on passive margins, only on a very small scale. So stuff coming down the hill slope here, you're going to have resting here. Also, this facilitates exchange, so if the river floods, there's actually possibility of perching stuff up on floodplains. So there's an exchange process going on too. So we have to factor that into the chemistry that we're going to see. Precipitation obviously is a major variable because that's what's driving the erosion. So what you see on the left is a timeline of daily precipitation over from 2014 to 2016. The little green bars are actually when we sampled our storm events. And I want to point out that we really sampled in two parts of the year. One was in June and July, which was really the rainy season in this area, but also it's when the surface is covered with vegetation. And then we got lucky that we got 2 October events. Up to October, we tend to have a fairly dry period. You can see the little gaps in there. But what's notable about October is that this is after harvest. So now the fields are not vegetated. In fact, they're quite bare. And so as you might imagine, we might see differences between those times of the year. The other thing that we were able to do is take radar data from NOAA and track the storm trajectories. Now, my initial assumption in all this, when I thought, oh, this is a very small watershed, one of the reasons for picking a tiny watershed is that when a storm comes through, the rain coverage should be roughly even, and we don't have to get into this trajectory business that you have to worry about with bigger watersheds. But as it turns out, we do. Okay, as anyone who lives anywhere, you know it could rain on your house and not on the neighbor's house. And so we can actually, you're going to see examples where we're going to use the signal that nature gave us with the trajectories to actually tease apart the storms, the storm response. So in this case, you can see that the yellow is more intense in terms of millimeters of year. So the upper part of the watershed got a lot more rain than the lower part of the watershed. And that actually has consequences. I should point out, most people would treat this as noise. But this is an opportunity. Okay, so what do? What we did was we put out auto samplers, collected samples every few hours when we saw a storm coming, about 700 milliliters a sample. Those samples were frozen immediately, but then eventually they were brought back in the lab and we filtered them. The dissolved stuff went to Purdue, and Tim Philly, who some of you might know, is working on the DOC component. What we kept the particulates. And what we're going to talk about today is some biomarker work done that we've done with GC mass spec, and some isotope ratio elemental analysis work where we have carbon isotopes, nitrogen isotopes. I won't show the nitrogen today, and POC concentrations. And we're just going to try to use this information to tease apart what is in a storm event. Okay. Your eyes tell you a lot. So we knew immediately when we start filtering the first samples in the lab that something exciting was going to be seen. The colors change through the storm event. So we have from time zero pre-storm up through the rising limb of the hydrograph, peak flow is up here, and then it falls back down. And you can actually see the coloration change. At the beginning of the storm, the very first sample or two, you could look with your eyes, you could see little chunks of algae in there. Not surprising for an agricultural stream that's eutrophied. You're going to find algae growing the stream. And then what we noticed is that as the hydrographs started going up, the discharge started increasing. We went from a light colored silt to these little dark aggregates that looked like surface soil particles. Very stable little aggregates that came on the front side of the hydrograph, and then they disappeared and we went back to this light colored silty material. Okay. What do we know are the sources out there? So other people have worked out there and done stable isotope partitioning, but I think you go out and use your eyes first before you throw anything into an instrument, and then you start using the instruments to help constrain things. And so what we know is that there are algae in the stream at least certain times of the year, late summer, early fall particularly. And we have isotope values, carbon and nitrogen, as well as biomarker analyses, and we can kind of place them over here. We have banks along the whole creek, which we know are going to be eroding, and I'll talk about that. We've collected a core of alluvial material right next to a bank. It was not exposed, and its isotope values fall in there. Tim Philly's done very extensive work on the row crop soils, and we know what they look like. They have a lot of C4 plant, corn signature in them. Prairie, C3 plant is there. No one's ever analyzed the particular organic carbon coming out of tile drains, and this being the Midwest, we have literally millions of hectares of land that are being drained so that the water moves off because it's so flat after a storm. You have to move it. So these tile drains are pumping out lots of water and lots of nutrients, but we don't know what kind of carbon. And it's isotopically quite weird stuff. It's there. That's a whole nother story. But what we see is that all of our river or stream samples are falling in between our supposed end members. So what that does is it gives us a little bit of confidence that we've captured maybe 90% of the signal with some of these sources. But what we don't know from this is how all these parts fit together. This is just a mishmash. And this, quite frankly, is mostly what you would get most people, if you were to look at the literature, is they go out and get some grab samples, and then they try to do isotope mass balance and try to parse all this together, figure out how much is algae, how much is that. You can't do it. There's too many degrees of freedom just doing it this way. Okay, so let's move and deal with the storm event. So this is in October 2015. It's one of our better storm events in terms of sampling continuity. This is in the upper reaches. So here's the precipitation. And you can see that it basically rained for a day. The discharge follows. And so, as typically happens, there's a lag between discharge and precipitation because the flow path the water has to take to get to the channel. And this is what the particulate organic carbon concentrations look like in the stream for this event. Not the pretty theoretical picture that we thought we would see. So what are we seeing? We've got this little bump, which probably continues out this way, pre-storm, before it actually starts raining, which, as you're going to learn, is the algae. It's as the system is sitting there and is in reactor mode, if you want to think of a stream that way, and the water's not moving, we have a lot of algal production. We get another peak, a much bigger peak and very dominant peak during the precipitation and on the rising limb that prior studies in this area have shown tend to be beryllium-7 rich. So we actually think, and as you're going to see, we'll show more evidence that this is a surface source of surface soils, we think. And then the main peak, the more ubiquitous peak that we see in every storm event, is the main sediment wave that's riding with the water. Ebbs and flows with the discharge. So we're actually going to look at these more closely to figure out what's in these things. Let's first, though, look at how this wave moves down, how these peaks move down through the system. So here's South Amana again, and so there's our 3 peaks. You can see the discharge. Again, this was one of these storm events where the precipitation was more or less even across the watershed, so it was a simplest case. Let's focus on this peak, our early flush peak. It's getting smaller as we go downstream. Okay? We think this is due to 2 consequences. One is the loss of connectivity. Remember how we went from a V shape to a box shape. If this is in fact a surface source coming off a field or something like that, it's going to be easier to get in the channel up in the uplands than it is down towards Coralville. And so we see that attenuation. Also, if it happens to be coming from a row crop field, we know those are more concentrated in the uplands too. So I think it's a combination of erodibility of the surface and the connectivity. The other thing to note is how the complexity of the system is changing. Discrete peaks here, some discrete peaks but getting broader, and then more peaks, but the whole thing's merging and getting broader still. And so what we think is happening is that you've got all these little tributaries that may be generating their own little South Amana type signal, but they're all coming in somewhat asynchronously and merging, and so that we actually have them all adding up and generating this more dispersed looking signal. It's a little more complicated than that, but we're building the story. So now we're going to add our isotope measurements, delta C13, as an indicator of changes. So remember, C4 plants corn are going to be up somewhere around -10, somewhere up here. C3 plants are down in this region. If we go to South Amana, what we see is a tremendous gradient, a quick response from C3 plant, in fact, very negative numbers, which we think are probably due to the algae themselves, to very reasonably positive numbers quickly with the early flush peak, which basically I think adds additional confirmation to the fact that we think this is coming off the bare cornfields in October. We see a similar type of response in Oxford, though the slope is a little different. And then we still see a peaking of some C4-ish material, but note it's coming much later. And we'll see why in a few minutes. And the slope's much gentler. So I want to come back to this issue of storm trajectories, because it's going to help us piece together what's actually happening in that big peak that we're getting with the main water wave. So if we look at this storm event in June 2016, what we note is that the rain is very light in the upper part of the watershed. It's blue, it's down here, and heavier downstream. And what should be no surprise is that what we see is a muted response in the upstream part of the watershed and a bigger response downstream. So what we're seeing primarily in this system when we look at the particulate organic carbon is organic carbon that's principally sourced more locally or proximally. It's not coming from downstream. If we flip the coin and look at a system where the rain was very heavy upstream and less so downstream, what we find is that there's a big time difference between the peaks, where normally we didn't see that. So the watershed's small enough that normally the peaks are all peaking very close to each other, maybe an hour different or so, but they're all very close because the whole watershed's getting some rain. But in this situation, it's so much heavier upstream that you're now seeing the contribution of the distal part downstream more clearly. In other words, it takes a certain amount of time to get from South Amana down to Coralville. So all of this sort of illustrates what I think is intuitively obvious to everybody, is that if you're sitting and sampling in a particular location, you've got two general sources of sediment and organic carbon. You have the proximal source that's coming from the local area, but you also have stuff coming from downstream. And now we're going to look at this in a different way again. So you can see we've spent a lot of time just pulling apart this onion, trying to figure out what all the layers are. And so instead of looking at organic carbon responses in terms of time, which is what we've been doing, we're going to look at it in terms of discharge, which the hydrologists like to do, particularly for dissolved species. I've never seen anyone do this for particulate organic carbon before, but it's very interesting. So you might expect that as discharge goes up, POC concentrations might go up because of increased erosion and carrying capacity. But these things are rarely perfectly linear in that typically you will have a hysteresis curve where the trajectory going up is different than the trajectory going down. In sediment, we typically see clockwise hysteresis, which is what we're seeing with the POC, which basically just means that the upslope has a higher concentration as the discharge is going up, and then as the discharge is going down, it has a lower concentration. And we can imagine a number of reasons why that's the case. The isotope values, on the other hand, are a totally fascinating story. Again, we see our isotope cycle going from light to heavy due to the corn. We see the slope changing as we move down slope. And then what was really fascinating is it's clockwise, clockwise, counterclockwise. What it means is that the corn signal from the uplands is arriving late, and it's arriving on the tail end of the hydrograph. We've already started to see hints of this. Okay, so we're going to put it all together after about this last slide. So I mentioned previously that we think the conductivity is decreasing between the hill slope and the channel as we move downstream. The other thing that we think is changing is that the bank erosion is increasing as we move downstream because the banks are just getting taller. There's more material, there's more alluvium being collected in the floodplains, for instance. And you see an example of what the banks look like here. And this is what you're looking at on the left side are data from a core of the alluvium, what we call post-settlement alluvium, which is material that has arrived since European settlement, so about 1850s, and they start doing their rather heavy-handed agriculture. And what the take-home message here is that we're recording quite an array of isotope values in this. This really is going to mess up the signal, if it will, or change the signal, because it's a mixture in itself. It's also aged material. This is old stuff compared to the surface. So the erosion of the banks, which we know is going on because we can look at photography over time and see the bank meandering, and there's been instrumentation put out to look at this. We know that this is basically going to lengthen the time scales that one has to consider if you're doing a carbon budget, if you want to look at carbon sources and sinks. This is going to shred the signal, as Jeremiah and Chris Paola have suggested. And this floodplain or bank source is going to become progressively more important the bigger the watershed gets. Okay. A few biomarkers. I know Tom is out there, and I thought I saw Liz. We have to have a few biomarker measurements that we're still trying to sort out. So there are things, so we've looked at a variety of lipids. What I'm showing here are concentrations, these dark bars on the bottom are the C16, C18 fatty acids, which are not diagnostic of much of anything other than life. I mean, they're the most abundant, ubiquitous fatty acids out there. But what's very notable is that their concentrations normalized to organic carbon are actually the highest at the beginning, which is when we see the algae. So basically, they're an indicator of anti-vascular plant material, which have low fatty acid compositions because of the cellulose and lignin. So you can see those numbers, and we have other sterols and things that I could have shown you on this. What's really notable is the lignin, which is coming from vascular plants and thus from the landscape, pops up with our POC peaks, and then it meanders around. We have a series of soil indicators. I don't think we fully understand where these things come from or how they're formed in soils. There's some ideas, but we can track those as well. And one of the things I just want to point out, we have no answer for this at all, is that the soil indicators tend to peak right on the leading edge of the POC peaks. So I don't know if we're seeing some hydrodynamic sorting or if there's something else going on in the landscape. There's a clue here to some detail of what's happening in this system that we haven't figured out yet, but we will. Okay, so to bring all this together, I realize this is a lot of different types of pieces of information, is if we go up in the upper reach, what we can identify is at certain times of the year, particularly in October, we can see a time series of responses of peaks. We can see an algal component that's there before the storm comes. We can see a surface soil component that we're fairly convinced is coming off the row crop fields based on the isotope values and the beryllium-7. And then we have this broad, more ubiquitous peak that is probably integrated material from upstream, but also includes some channel erosion. As the signal moves downstream over this relatively short distance, what we see is that the local source is actually decreasing. The local surface source is becoming less important. And the integrated peak coming from upstream is becoming more important and more complicated. So much so that we're still trying to figure out how do we tease it apart. But we have some clues. So for instance, we can find evidence that this peak is now over here, coming out late. So we know we have to interpret these peaks in terms of local sources and upstream sources. And so that's what we're currently trying to do, is figure out some way using this network of samples with a variety of biomarkers and other indicators to try to really dissect what's in this peak. So that's our signal on a small scale. Okay, so we're going to very quickly go to the larger scale to sort of ask the question, like, once you generate this material, where in the world does it go? And again, we're going to come back to this picture of the 3 traps on land that Mead proposed. What's rather fascinating is that there's all kinds of sediment budgets out there for these 3 types of depositional environments, these sinks. But there's never been a study done that we can find where all 3 traps were in the same watershed. And so we could look at their relative importance, and then we could look at the organic carbon in them. So you're going to see it today. Okay. So first, just to give you sort of a global overview, if you go and data mine what's out there, what do people find? Is that the flux of particulate carbon from water erosion, soil erosion, is somewhere between 4 and 6 petagrams per year. If we look at the colluvium and alluvium deposition, which would be at the foot of hill slopes and the floodplains combined, we can see that the bulk of the material is being trapped there. In fact, the mantra that you read when you read these landscape papers, the erosion papers, they always say the carbon doesn't travel very far. But the thing is, they never tell you how far is far. And that's really what we're trying to do here, is how far is far? A kilometer, 10 meters. Lake and reservoir trapping, you can see quite a range, because there's actually quite a range of different types of rivers and of lakes and reservoirs, but about 10%, 9%. And so here's the number, not unlike what Bob Mead came up with. 8% of the eroded material is making it out to the ocean. So it's a leak. So what we did was in Sangamon, which is in Illinois, which was the other CZO site that we studied, we have a reservoir, which is down at the terminus of the watershed. We have floodplains that we've studied, as well as depressions that are left over from the last glaciation that we think are traps, which are basically at the base of the hill slopes. The whole system's mostly corn and soybeans, so we have our corn signal again. So let me just go through this very quickly to show you what we did. Lake Decatur, I think, is our best constrained estimate of carbon budgets because it has a defined footprint. We know the river was dammed in 1922. The city rapidly became concerned about the lake infilling because of all the agriculture and the high rates of erosion. So they commissioned sediment studies almost every decade up until the 80s to figure out how much infilling is going on. So we have a really good sense of accumulation rate up to that point. We've also done cesium-137 and lead-210 to get a more contemporary accumulation rate. And we've done organic carbon measurements, isotope measurements, biomarker measurements, as you'll see on this. So we have carbon budgets for this nailed, I think. I think the post-settlement alluvium, which is what we're interested in, that sedimentation that's occurred since we started heavy-duty agriculture, is also well defined in this area by Dave Grimley, who's down at NC State or visiting in North Carolina these days, and Allison Anders. And what Dave did was, among other things, using lead-210, but also using magnetic fly ash, which is produced from coal combustion, which is more or less contemporaneous with the agriculture. By looking down core in the floodplains and finding those areas where you had no fly ash, you basically had identified around 1840s, 1850s. And so you know how much has accumulated since then. And what they found was a very nice relationship between this post-settlement alluvium thickness cover and valley width, which then allowed us, now we have a proxy to get at volume of this accumulation just by using GIS. You just go and measure valley width and you can estimate what the volume of post-settlement alluvium is. And of course, when you measure organic carbon and do your other measurements, we have that information as well. So we have a very good inventory for that. Our fuzzy inventory are these depressions, which I get, as I said before, they're from a glaciated landscape, leftover, they're filling in, trapping sediment. We identify them by the soil type, some very poorly drained soils down at the base of the foothills. We don't have as many post-settlement alluvium measurements and they kind of range around. We don't have correlations with X or Y. So all we did was we took sort of a nominal number, 10 plus or minus 5 centimeters for that, and took an average organic carbon measurement for soil surveys for these types of soils and came up with that budget. And so here's what we have. What we find is that with big error bars on some of these, about 60% of the organic carbon we're finding is in these depressions. A third of it in floodplains, 6% in Lake Decatur, and we know how effective Lake Decatur is at trapping sediment, so there's about 2% leaving the system. When we add up all that sediment and then put it back up on the landscape from where we think it eroded, we can calculate a thickness, a soil thickness. We can then estimate how much carbon was in that. And so this is our estimate of what was actually being eroded from the surface, back calculated. But what's the take-home message here is pretty obvious, is that stuff drops out when it can. Okay, not exactly rocket science, but it kind of tells us that. A brief word about our sedimentary record, because we have to do a little homage to sinks a little bit, is that we, of course, cored Lake Decatur and did biomarker and isotope measurements with an idea of trying to reconstruct the history of deposition in the lake and what was happening on the landscape. And what we see is that there are peaks in Del C13 that we're fairly certain are due to the row crop inputs. They coincide with cellulose and lignin peaks, so it's definitely a vascular plant material. What we found rather interesting is that, I should mention, I did something I would never normally do, is put a time scale on our core data as opposed to depth. And it's only because we have such confidence in our geochronology here. We've been able to show that we're good within 5 years by different methods. And here's another indicator that we're probably right. So if we take the annual precipitation in the area, in the watershed, it's hard to see what could be generating these signals. If we do a 5-year average, a running average of the precipitation, all of a sudden we see that there are wet periods that seem to be coinciding very nicely with our peaks. And so what I think is happening, it's telling us that the system, it's very hard to get something to move a particle to move a great distance in one storm, unless it's a humongously huge storm. That in sort of the normal annual basis, things are hopscopping across the surface. And they're going to move faster if it's a rainier time. Okay, so to wrap things up, points to consider. This is just a watershed in the Midwest. Can we learn broader lessons? One, if you had a system that was not glaciated, or it's been much longer since glaciation and those depressions have filled in, then you would expect better conductivity down to the channel. Okay? And so floodplains will become more important. Where you put a reservoir is also important. If you put the reservoir up in the upland, it's going to receive most of the sediment and starve the floodplain. And we have examples in the literature where that's happening. And then if we compare this to the steep slope, small mountainous rivers, clearly we have much better trapping efficiency in these than in the small coastal systems. So anyway, conclusions and implications of all this. We can identify a temporal sequence if we look hard enough in a small enough spatial scale of sources, such as in-channel sources like algae, surface soils that come in with the rain, and then the main peak that rides the hydrograph. Some of these sources are conditional, some aren't. That temporal sequence is very rapidly lost as we start adding in tributaries. Floodplain sources or bank sources, which people have called signal threaders in the signal propagation field, are going to become much more important the bigger the system, the longer it reach, to the point where at some point they become the signal. They mask the upland signal. And I would add, just as an aside, for those of you working in marine systems, we have a mechanism now for our pre-aged terrestrial carbon. It's this material that's going off the side. And we also can explain some of our terrestrial C14 reservoir age that we can get on stuff by storing it in floodplains and the like. And then finally, if we want to extrapolate back out globally, the export efficiency of the POC is highly dependent on topographic gradient. The low gradient systems, what they're doing is they're leaking out POC that is not going to be representative of the whole landscape. In fact, they're really more representative of the storage, I think the subsurface storage of the landscape. So I'll end there. And I'd like to thank the IML CZO team, of which there were many people who participated in this. Thanks for listening.
Great. Thank you, Neal. And because you are well known about your earlier work on the Amazon shelf, Papua New Guinea and New Zealand. So, but now suddenly you shifted to the source to help us understand the watershed, how much the POC, the signature. So this is great. So go ahead. If you have any question, you can unmute yourself and go ahead to ask. For the audience on our YouTube channel, if you are able to shift the Zoom, that would be great. If not, you can type your question on the chat over there. I can read your question for you. So go ahead. And so Tom, go ahead. And then next, David Master.
Okay, thank you. Neal, as always, very careful and thought-provoking in the work that you've always done. But I have a comment and a quick question. One is, because I haven't read this work yet, but it seems to me that the kind of detail you have on this system allows for a certain integration into some of the sort of dogmatic limnological work, like the river continuum concept and Pete Raymond's more recent pulse shunt kind of work. And I don't know if you get into the discussion of that in your papers, but it seems like this is like really perfect for adding or integrating into those concepts. David Buttman is another person that was one of Pete's students that's at UW. So I see you shaking your head, and I figured you would know who those are. So that's just sort of a comment on that. The quick question is, it looks like the nitrogen coming out of some of those tile drains might have some nitrogen fixation going on with some of the numbers. And I don't know how else you get those numbers down that low.
Yeah. First, yes, I've had one of my big challenges to moving to the Midwest and getting in this is to get into another community. And as any of you who've tried to change fields, it's a little bumpy. And so the hydrologists are as unforgiving of us as as marine scientists have been of them when they try to go into our realm. Sometimes we bust people's chops. So my chops have been busted a few times. I've got bruises. But anyway, yes, we've had to get into this. And the whole pulse shunt thing is rather interesting because they're starting to make predictions that the chemistry will change in some way. In fact, what they tend to see without good explanation is that early on they see an aliphatic rich signal that becomes aromatic, but they kind of wave their hands about it because they haven't done the molecular work. And I think we can nail it exactly. We know the aliphatic, as you know, is going to be the algal signal when the system is sitting still, sort of this reactor pipe conundrum. And then when it kicks into gear, we're getting the terrestrial, more lignin-rich material. So we can actually just check the boxes of a lot of these ideas. The tile drain stuff is quite a fascinating situation, and it's one. We have two possible solutions or explanations. One, we're looking at very old soils that are Los base, and we're looking at the remnants of the organics in those. And there's just been diagenetic isotope effects and stuff that have driven the isotopes in that way. That's the unexciting mechanism, but we have to consider it. The other mechanism is, and this is the one I love, and I just need to get a microbiologist out here to study, is that the fact that we are putting these pipes in the subsurface, we're creating these artificial caves where oxygen's coming into the subsurface and starting to oxidize the organics and stuff. And you would expect maybe a chemolithotrophic group of organisms, bacteria, to kind of set up shop at this interface in the tile drain. A whole nother ecosystem of microbes, which would include some nitrogen fixers, but nitrifiers that were autotrophic, that would give you these very light carbon isotope numbers, as well as the light nitrogen isotope numbers. And you can find culture experiments where you can see these types of numbers, but we just haven't done the microbial work to show that. But that would be the exciting explanation.
Thanks, Neal.
Hey, Dave.
Thanks, Neal. That's fabulous stuff. Whoever thought that 20 years ago when we were doing SlopeX together that you'd be looking at cornfields and soybean fields and finding out waves of small rivers carrying POC.
I would have bet
anyway,
my last C13 diatom that I would not have been.
Yeah. My question is, do you have any grain size data from your small river study? And have you done any carbon-14 on any of the storm-driven POC transport? And the reason I ask is, you remember being an old Amazon person, John Hedges had the interesting finding that the coarse fraction was kind of fresh leaves and stuff that had a bomb C14 signal in it. And then the fine fraction was more old soil alluvium that was 1,000 years old coming out of the Amazon in the fine fraction. So I'm wondering if you consider seeing, using that kind of tracer.
No and no. I mean, we have our great, first of all, we have to, one of the challenges I had for this project was I was given, you're going to get 1 liter water samples and you've got to do everything on it.
Yeah.
So basically our lab filtered everything, and then we were sending this stuff out to 4 different labs for analyses. So I had to basically get my infrared spectroscopy, my EA isotopes, and biomarkers all on one filter, and then share whatever I had left over, which really isn't much. So carbon-14, we could probably do it on a few samples at peak discharge. But I just had, we've been so busy doing the biomarker stuff and trying to make sure that that works well, that I just haven't had the bandwidth to even think about carbon-14, or the budget for that matter. Grain size would be very interesting. All we have is really visual. We look at the stuff under a microscope, but we haven't quantified it in terms of how much sand, silt, clay in there. But you can see the differences through the storm. Definitely gets coarser as the velocity goes up, as you would expect.
That brings another question, and that is, what if you compared a system like the Midwest, where you might have more glaciated, silty soils, as compared to your old stomping grounds in North Carolina, where most of the soils are Triassic red clays that are much finer and have much more iron and have kind of a different cohesive nature to them than much of the more organic rich soil in the Midwest. How do you think they'd weather or respond to these pulses in rainfall differently?
Yeah, I kind of wonder if you would actually see such prominent early flush peaks in North Carolina. That would be a very interesting experiment. People see them. These early flush peaks are actually a very common observation in rivers and streams. But I've never thought about looking at the lithology or the soil types or grain size around the world to see if there's any correlation. That would be a very interesting data mining exercise to see where you see these early flush peaks predominantly. Obviously, you need a very erodible surface.
Thanks.
That's a really interesting question, Dave. I have to think about that.
Okay, cool. Anybody else? If you do have a question, you can unmute yourself, Bob.
Yeah, I have a question, Neal. Well, first of all, that was fantastic as usual. So I'll just pile on that accolade.
Oh, thank you.
I'm curious as to what you think. You described the nature of the signal that's generated in events in different parts of I'm thinking particularly of the Iowa system. And I'm wondering about, since you monitored in June and July, what you think the compositional changes might be if you had looked in early spring before you had crop cover and perhaps after a tilling event, the relative timing of human disturbance versus natural seasonality of plant growth versus when you get a precipitation event.
Yeah. The short answer is I don't know. I could make some idle hypotheses, I suppose. My grad student, June, is literally analyzing our first and only spring event now. So in 6 months or less, we'll probably be able to answer that question. The fields are bare in spring, but they're often really wet. In fact, this is one of the challenges, as if you watch the news, of farmers getting out and getting their equipment out in the spring because they have to wait for it to dry, and it's hard sometimes because it's the rainy part of the season. So whether supposedly the erosion, this carbon yields or the sediment yields can be quite high in the spring if you catch it at just the right time, based on the studies that have been done. It's going to be rather degraded material, I would think, because what the farmers do in the fall actually is after they harvest, sometime in October or November, they till then because the fields are dry. And so they till all the stover down into the field. And there's presumably a great deal of degradation because the fields aren't swimming in all the stover when you get it back out in the spring. So even though it's been cold, that stuff's been degrading. And then they go out and try to do a light till if they have the opportunity in the spring, but they try to minimize all the heavy work in the drier fall. I would hope that we would see more highly degraded material. One of the things that we're looking at are like the lignin acetaldehyde ratios in combination with these soil indicators to try to get at degradation indexes, to really kind of get a sense of surface versus subsurface soil. But you raise a very interesting question because that index may change seasonally as well. The other thing that we note in our data, and I didn't have time to get into it, is that the response to a certain amount of precipitation changes seasonally. So in other words, you get a lot of discharge in October per millimeter of rain because there's nothing to intercept it. In June, July, the transpiration rates are really high in cornfields, some of the highest. And so you don't get as much discharge out. And so the erosion is much less. And so what we've been able to do is by having this radar data where we can get localized rain for the different subcatchments. I didn't show it to you guys, but I actually can show you that carbon yield, how much carbon you get per square kilometer, varies as a function of the storm intensity across all of these. And there is some noise in there from the season, but the season doesn't override the master variable, which is just rain. A meandering answer to your question because I don't have a good answer.
Thanks, Neal. It's great.
Okay, so if any more questions?
I have one more.
Okay, Tom.
I'll be quick. Don't
be quick, Tom, because I have to go out and shovel 2 feet of snow.
Okay. Well, gee, I'm glad I'm in Florida.
This is a great Great
I'm
parade.
glad I'm in Florida, maybe feeling that for the first time in a while. But yeah, I think your comment about bringing in sort of a micro person is microbial person is something that I've been thinking about a lot in recent years in terms of uh, sort of where the biogeochemistry is going in the 21st century. And I think it seems like the open water ocean people have been on this much more than sort of the source to sink. The source to sink group seems to be much more geochemically driven in the way they think about things. But I think when you look at these gradients that you're talking about in a nice, well-controlled small system like this, and you sort of expand that to much larger river systems from the Andes, let's say, to the mouth of the Amazon. I think something that's really still missing in a lot of these conversations and talks is the importance of natural selection in sort of rebooting the microbial communities along these gradients. While we tend to think about hydrology, mineralogy, erosion, all of those things along these gradients, the thing that's incredibly important now in the Anthropocene is that we've changed the selective gradients by damming and all these other artificial structures, as well as climate change. And so my point is that I think while microbes have been mentioned many times by people, I still think that the sort of aquatic critical zones where the processing rate and the kinetics are changing needs to be incorporated much more than the way source to sink people typically think about what goes in the pipe and comes out the other end.
Yeah, they're a very dynamic. I hesitate to say tracer because in some ways they're too dynamic. But there's been a little bit of microbial work done in our CZO and where they've tried to sort of do a source to sink microbial study. And in a way, the result was conclusion was rather unremarkable in that everything's connected. Yes, the microbes communities adjust to their particular environment, but when it comes down to it, everybody's everywhere to some extent. And so I think there's got to be some refinement of not only the way we think, but also the way that microbial people are thinking too. I don't think they've landed on the really important questions because everything's so new, we don't know what to ask yet. I think in many ways. And I think there's a very fascinating opportunity to start melding these different approaches, as you're suggesting, to see what the next generation question is.
You're absolutely right on that. There's still a lot of people stuck in the who's there mode and sort of having their list of bacteria, but there's a whole new group that has now looked at more nitrogen-based, carbon-based process metabolism.
Great. So if you know any other comments, questions, I think maybe we can stop here. So thank you, Neal, very much. And
thank you.
I'm sorry you had to go out to dig that 2 feet snow, and at least you are lucky than our colleague in Texas. So thank you for all the audience for coming here. And as a reminder, this coming Friday, Hans Paul will give us a very interesting talk in our North Carolina, coastal and river system. So please come back this Friday. And so thank you very much. And so have a good day.